Hello and welcome to Future Medicine AI. My name's Harry, I'm the editor here, and today I'm bringing you an interview with Ofra Benny, who is a researcher leading some incredible work to revolutionise the way that cancer care and treatment is delivered to patients. So essentially, her and her team have developed a new method for cancer cell classification, and it works using a combination of machine learning and flow cytometry by exposing the cancer cells to different size fluorescent particulates and measuring their ability to uptake these different size particulates. They can essentially use machine learning to classify the cancer cells. And the implications of this are incredible and very powerful for clinical practice. And they're already working on clinical trials which basically allow clinicians to take cancer cells from patients and apply the new algorithm and method that can predict what treatments are going to work best, what ones won't, and whether they'll recur, whether they will experience recurrence. So it's extremely powerful, and the story behind it is even more fascinating. And there is a beautiful arc of discoveries that led to the achievement that Ofra and her team have reached at this point, which is an extremely high accuracy classification of cancer cells that visually appear indistinguishable. So this interview is really interesting, and she gets into her background, what led to this discovery, and her other endeavours and what the future holds. We talk about the AI algorithms and the training. It's really interesting. So I hope you enjoy, and yeah, catch you next time.
So I'm Ofra Benny, professor in pharmaceutical sciences here at the Hebrew University in Jerusalem. I'm coming from, actually I did all my degrees, I did it at the Technion in Israel, which is kind of MIT. I would say that we have like the equivalent of MIT. So I'm coming from engineering. And then I did my postdoc. I started as biotechnology engineer, and then I went to a long postdoc at Harvard Medical School. I did my postdoc in a very well-known lab of Judah Folkman. He was very well-known scientist and researcher, and unfortunately he passed away when I was there. I was his last postdoc.
Oh wow.
But yeah, it was a unique experience.
Yeah, I can imagine.
Yeah, but luckily, I had the chance to work with someone like him who was giant, it was very creative. And I came from engineering and materials, and then I started to dive in the field of cancer research, cancer biology. And all over my career, I'm kind of integrating these two disciplines. So I always have engineering, have biology or biological sciences. And recently, with the emergence of all the AI tools and the machine learning, we realize how much it can contribute to our field of biology and cancer research. And gradually, we started to have more and more projects that involving AI. So we just give a background about my lab here. So I'm running a big lab, relatively 15 students and team members that focus on bioengineering and integration of biology and engineering. That's more or less what we do. And all our projects are aimed to address really unmet need, like clinical unmet needs. And one of the advantages that we have here in this atmosphere that we have specifically in the university, in my campus, is that the preclinical side, which is the research side, we're physically very close to a hospital. So we are communicated a lot with clinicians. And in
That's
that
very
way,
interesting.
it's really important because
Yeah.
we know we address real questions and not
Yeah.
like something we make in our own heads. So this project started a long time ago when we realized that in cancer, much of the selection of the therapy that is given to specific patients many of these decisions are based on trial and error, kind of, or on fixed protocols. So basically, a patient comes and diagnosed with a certain cancer, and based on the long experience, they give him protocols, him or her, very fixed protocols for first-line therapy. And then in many cases, the disease come back and reoccur, and the patients cannot really benefit from the therapy anymore. And then the oncologists need to give them something new, but they have no idea what to give them sometimes. So today, as you probably know, there are many advances in genomics, for example, so they can have genomic profile of the patients, they can find specific mutations that give them some ideas of what therapy to select. But in most cases, there is no enough information, even in the genetic analysis, that can help the oncologist to give really a rational selection of drug, of therapy. So we realized that this is a big problem. Eventually, we want to give the patients the most, personalized therapy, the best optimized therapy for the specific person. And we understand that there is not enough information that can be translated to the real life, because genomics or transcriptomics, or all the omics, particularly proteomics, all this, because you come from this field. So it's highly complex, especially when you start to analyze single cell. So we started to say, wait a minute, so we need to find some more general principle for predictions, for trying to predict how the cancer cell will behave. So we wanted to be able to measure something that is already a result of many parameters. And we had, at the same time, we had a big research in my lab that we found very interesting correlation between cell mechanical properties, meaning the stiffness of cancer cells, and their ability to be aggressive or less aggressive. So we said, okay, so this is very interesting. So maybe we can just measure mechanical properties of cells. Let's say you can squeeze cells by AFM or whatever, and you can say whether they are very stiff or very deformable. So that was another paper that we published also in the Science Advances 2 years ago. And we found that indeed there is a correlation between mechanical properties and behavior or function of cells. For example, we found, it's very interesting. So the cancer cells that are more deformable, they are also more aggressive, which makes sense because they know how to move. So this is one aspect. That's nice, but to be able to measure that, this is extremely difficult because now you have to squeeze each cell. It doesn't make sense. But then another big advances that we did, and that's really what we had in this project, is to use actually a kind of a proxy measurement for that. So we can use particles, solid particles, nano or microparticles. And we found that actually the uptake of these particles are really dependent on the ability of cells to make these deformations. So it gives us
Is
for
that
the
to do with the membrane being able to take in?
Exactly. So imagine that you have some fluid like balloon that has to engulf something, so it has to change shape. So one aspect is the fluidity of the membrane, and the second aspect is actually the formation. It's all the skeleton has to really change shape. So we said, okay, so we want to measure both. I mean, it's interesting to see also the fluidity of the membrane and the ability to deform. So what we've done, and this is really interesting, so we took a panel of particles. Each one is coded with a unique color. Okay, so we have, let's say, small nanoparticles are red, medium particles are green, and the large particles are blue, and it's always like that. So now we take these particles and we incubate them with cancer cells. And now we started to measure the spectral signal, the colors, basically multispectral. And then by looking at the colors, you can know how much of the particles were taken up by the cells. It's really nice. Hard to explain it to sometimes all the rationale, but that's how it worked really. It was a journey that we started with understanding the mechanical properties, and we realized that uptake can be a proxy measurement. And now this is the huge step that we did now is that really by looking at patterns of uptake, and now the machine learning was so useful for us, we can actually link between uptake and function. So what we've done was to start to do the learning phase, the training. So we did a lot of analysis. We took one population of cells that we knew already that is more aggressive than other, because we did this, it's a kind of bias approach here at the training, because it's supervised basically. So we index the cells. We said this population is very aggressive because we measured that, and this one is not aggressive. Now I want you to look at all the uptake patterns, but in a single cell. We use FACS. FACS is you probably know, flow cytometry. So we can look at the single cell on these uptake patterns. And then we did the same thing with resistant to certain drug. So we looked at drug resistant, we look at aggressive invasiveness of cells. And after the training session, then we could give again a mixed population and ask the machine, now that I give you only uptake information, now you have to tell me how much of these cells are really, aggressive, how much they're. So this is the point of this study. It's very nice. We got very high accuracies.
You really
And
did.
I think, yeah, no, it was very accurate. And I want to say something that is very critical here, because one of the controls that we did, we said, okay, but basically if the machine is so smart, maybe we can just let it look at the cells, look at the morphology, or because if function is so linked to physics, to the physical properties of the cells, maybe we can just give the size, the granularity or whatever. But then we took a subpopulation of cells that look alike. They look very similar. But we knew that one subset is more invasive and the second one is non-invasive. But they look very similar physically. And then we did the same test. We put these particles, we looked at the uptake patterns, and then it was able to find it. But if you would look at them, you wouldn't see this differentiation.
I don't want to take away from the really interesting thing that you've achieved, but just to talk a little bit about how the AI has impacted the use of machine learning, how that has impacted your research here. Because I actually had a meeting just before this with someone else doing some research, and I think an interesting point of conversation is how in academia you guys access the machine learning, and particularly the compute that you need to be able to achieve this. I think there seems to be a lot of people struggling to access in academia, because all of these big companies are used, like, they have loads of access to it, but you guys, I'm interested to know what your experience has been with that. And
Sure. No, it's a good point, because this is an emerging, it's an emerging technology for us, and I'm sure that the impact will be super valuable for medicine and for biology. And we understood, at my, I'm not coming from this area, but I recognize how critical it is, because I knew there are patterns that we cannot see in analytical way. I mean, it's statistical method. So I was sure of that. And then I knew that I have to do this link with experts in AI. So I think in academia, it's very important to collaborate at this stage, because it's very hard to be an expert on everything. That's the situation. So I think today, if you ask about the impact is for sure is revolutionary. I mean, it's going to change the medicine and human health, and I'm sure of that. And we already start to see the initial, we start to glimpse on to see the beginning of all that with the genomics and with the even drug design, all this. And it gives us an open to start with personalized medicine. I mean, it's amazing. We couldn't do this study otherwise because we had so much. The big data is really big at a single cell, and I'm sure there will be a lot of this in the coming years. But at this point, the access to have the accessibility to this, I think it's through collaborations for now, at least in the in the academic setting. That's what I feel. And yeah, because you have to defend your work somehow. I'm not expert in all, so I have to have someone that will be able to lead this and to train the students.
Yeah, I think that's really interesting, and I think you've hit the nail on the head there. And it's kind of like, it kind of builds on the point that people say like AI isn't going to replace people. People who use it are gonna replace people who don't in a way. Um, and and it sounds like it has empowered your research, and I think is it exciting for you to be able to have this new tool in your box that you can accelerate your research or do new things with this idea that you've got? Is it something that's exciting for you?
Yeah, no, I feel that this is a very critical tool that
Mm-hmm.
we cannot, if for now we used to be very kind of conservative with classical methods that we use for many years as researchers coming from biology. You have the statistic that but I feel that we have to open our mind now because this is another era. We have to be there. I mean,
Yeah,
and
of course.
I think we start to see that. I mean, all the innovations now involve big data.
Yeah.
I don't think, as you mentioned, yeah, people think that it will replace the researchers eventually, maybe in a long, long run.
Yeah.
But I think in the coming years, we will need still
Of
to generate the data to ask the questions. But I'm not ruling out, by the way. Might be that one day the machine will beat us. It might be.
No, I think it's so interesting to talk to someone like you about, because I'm interested in both sides of it. And I don't think AI is the solution to everything by any means. I'm not trying to say like, oh, it's gonna, hopefully it will solve a lot of our problems, but it's really interesting just to see how it's been used. And that's what I want to share, like through this platform that we've got. I want to share with people, how is this stuff being used, and hopefully give other researchers inspiration about how they can use it in their own work. So can you tell me a bit about the techniques that you use for the
No, we had a debate whether to use imaging indeed or to go to other types of data because we could do imaging. Imaging were, in our case, it was limiting for us
mm-hmm.
actually to use imaging because we are relying our our detecting measurement is fluorescence, and then in imaging sometimes you have overlays. It's technically quite difficult to do, so we decided to go with some other technology, which is flow cytometry. So basically in flow cytometry what we can do is to measure fluorescence, but it gives you the fluorescent in a couple of colors you can do at the same time, which is important. So you have a spectral measurement. But the importance of flow cytometry is that it can give it to you in a single cell and in large numbers. So potentially we could do imaging, but we decided to go with flow cytometry because of the throughput. It's relatively simple to do. You don't have to use a microscope. You don't have issues of overlay between colors and so on. So in our case, it was more making it makes sense to do it by flow cytometry, but definitely imaging is an option. I mean, we could do a lot of microscopic imaging and then to do machine learning, like image analysis. So
mm.
yeah, I mean, potentially it could work by imaging as well. but we decided to focus on this flow cytometry. It's a single cell. It's basically I was trying to coin this term in the paper that I call it mechanomics, because it's kind of a new types of omics. It's not genomics, it's a phenotypic omics. So actually I'm able now to give a lot of information in a single cell, like multiplex kind of, but this is already a phenotype and not specific. It's hard to explain what I mean here, but it's not the Lego, the each Lego part. It's actually already an emergent phenomena that you are measuring. So it's an interesting concept that you can look at phenotypes, and indeed in imaging you also can look at phenotypes because you see behavior of the whole organism or something that is sum of a lot of parameters. You
Mm.
already measure something that sum up a lot of factors.
Yeah, I think mechanomics is a great term as well. I think it fits very nicely what you've described in your work, and I guess that leads on quite nicely to what do you feel the implications of this, what you found are moving forward in terms of not just your research but potentially clinically as well.
Yeah. Yeah, so eventually what we aim to do is to take it to the clinic. So we have already a plan to start clinical validation. So what we do is we take biopsies from patients, from cancer patients. We do the same analysis, and now we have to wait and correlate it with the clinical outcome. So for example, we're looking at recurrence. This is one parameter, so disease recurrence. Let's say the patient is being treated and you can predict whether this patient will respond for a long time or maybe the disease will recur. And also I want to be able to predict resistance before they even start treatment. So this is what we are trying to do. I will mention here that we already have a spin-off company that's called Precure that is working on organ on a chip, if you know this technology.
I'm very interested now as well. That's yeah.
It's really
Please
cool
continue.
technology. So basically, this company, what we do is we take biopsies from patients here from Hadassah Hospital in Israel. We grow the biopsy, I mean the samples. We grow them, but instead of one tumor, now we do like hundreds of them. So you can screen drugs, and I can give the oncologist, we can give them an answer whether their a short list of drugs that are debating what drug to give the patient. We can tell them this one works, this one is the patient is resistant. So it's now in clinical trials, and so
Really?
actually I think it's really cool. So I think we will be able to integrate this technology with the mechanomics. Maybe next step will be to somehow to merge it with this organ on a chip technology, and then it's really personalized medicine. So you
Yeah.
take samples, you do the same analysis on a specific person, you can ask specific questions, but of course the training will have to be very specific for a clinical and like a narrow clinical question, for example, resistance or invasion or something like that.
Yeah. Wow. That's a really powerful implication of what you're doing. And what's your involvement with this? This are they associated with the university? Are they spin out? Did you say they were a spin out of the university?
Yeah, actually, I'm the CSO.
CSO.
Yeah, I'm a professor here. I have already 4 startups that I started for my lab. Actually, each one is in another, each one is like another field. Like one is a drug for ophthalmology, the second is for
Oh,
cancer.
ophthalmology.
It's a compound for AMD, for wet AMD,
Okay.
macular degeneration.
So you
Yeah,
have a
so
compound for it.
yeah, we have something very exciting. It's unrelated to this
Yeah.
conversation, but it's anti-angiogenic drug that is not dependent on VEGF. So
Oh.
yeah, so it's really nice because It's a new mechanism. We think we will be able to combine it or to use it for patients that has only partial response when they finish the Lucentis or Avastin or whatever. All the drugs today target VEGF, right, in the
Yeah.
AMD, and this is completely new mechanism of
Yeah.
action. So I think it's very promising, and we are working also with clinician with the head of the ophthalmology department here. It's a very nice. I think it's promising drug.
Your research or your work sort of spans across domains then, so you're not just focused on cancer?
No, not only on cancer. Most of my work is cancer or
Yeah,
angiogenesis, like the formation of blood vessels, and this is relevant for other diseases,
of course.
I mean, in ophthalmology, in the skin. So, but the motivation is usually we start with the unmet need, like we try to understand what is missing now, what solution needed to be solved using technologies and advanced materials and so on. And then we try to really tailor the solution per the problem. So we have medical devices. For example, we developed some drug-eluting devices, kind of implants. Many solutions that are half engineering, half biology, but it's always in a context of clinical issue that we are trying to solve. So that's more or less what my lab do. I mean, we have a big panel of projects that almost all of them are very applicable projects, but sometimes we have also basic science. I mean, in this case, for example, we started with the really basic research that we realized how the mechanical properties affect function, and that was very interesting. But then it led us to this application.
Yeah. It's a very neat curve of it. Sounds almost like I'm not saying you got lucky, but it sounds almost lucky that you found this relationship, because it's this is the kind of thing that sometimes there seems to be gaps of years between someone finding the
That's
mechanics and
true.
then like the uptake as well. These two things being
I
related.
know.
So yeah, that's really interesting, and yeah, I mean, thank you so much for taking the time to talk to me.
Thank you. It was great to talk with you. Thank you.
Thank you
Bye
very
bye.
much. Have a good day.
You too. Bye bye.
Bye.